Okay, in this video tutorial we're going to briefly introduce bacteria. So, you may have found that term a little bit confusing, bacteria or bacterium. Bacteria means many, is the plural, and bacterium is one. So if you've got one single one, we're talking about a bacterium. So we're going to start by looking at some of the parts of a bacteria, a standard bacteria. And remember, they do come in different shapes and sizes, but these are the generalized, these are the things you'd generally find in a bacterium, or that they may have. So starting off with this bit in here, DNA. Bacteria always have a circular piece of DNA, which is called, which we sometimes refer to as their chromosome. So you may see this labeled as DNA, and in some other diagrams it might be labeled chromosome. So their DNA or chromosome is a large circular loop, but it's often, because it's relatively big, folded over itself. So you can see this is one large loop, but it's kind of been twisted and folded in on itself. So the bacteria's chromosome or DNA is basically the information it needs to do all of the things it does. So it has the code or the recipe to make more bacteria, to grow and divide, as well as to produce the things that help it function and stay alive inside the cell. So it's the DNA is its genetic material. It encodes the proteins, and it's the function of those proteins that help keep the bacterial cell alive. So the DNA is kind of the sometimes it's referred to as the control center of the bacteria. It essentially contains all the information that's needed to keep it functioning and alive. If we look at these red spots, these are what we call ribosomes, and they're found in all living cells, and they're involved in reading the genetic code and making proteins. So proteins, if you're wondering what they are basically the moving working parts inside the cell, almost like the gears and cogs. Or if you think of the cell as like an office, the proteins are like the workers inside that office. They're large chemical molecules that essentially can change shape and interact with other chemical groups. And so some of them are enzymes, they speed up reactions. Some of them act as receptors and are involved in signaling and things like that. Some of them are structural, hold certain things in place. But essentially the DNA or the chromosome contains the recipes, the genes, for those proteins. And the ribosomes are what essentially read those recipes, the genes, to actually make those proteins. And it's the proteins that carry out all the different functions inside the cell and make it do what it does, or keep it alive. So next, I guess we could look at the moving from the inside out. We've got the cytoplasm is just the fluid jelly-like substance. So the cytoplasm is essentially the gel-like fluid that fills the inside of the bacterium, including all the things that are floating in it, like the DNA and the ribosomes. So it's kind of like, cytoplasm is another kind of term for kind of cell contents. Then there's the plasma membrane, this green layer. So the plasma membrane is just a thin layer that basically controls what moves in and out of the cell. So especially things like water and ions, like salts and things like that, it controls what moves into and out of the cell. So the plasma membrane is kind of the gatekeeper. It controls what's moving in and out. Then moving slightly out from that, we've got a cell wall. So the cell wall is what actually gives it its structural strength and kind of integrity. So these are kind of like, and the cell wall is kind of a fibrous network. So think of it like bars. There are gaps between them, so things can move in between the cell wall. It's more like a cage that surrounds the membrane and gives the bacterium, gives the surface its strength, so it doesn't get ripped open or break apart. So the cell wall provides structural integrity or helps the cell maintain its shape. And moving slightly further out from that, we've got a capsule, or sometimes this is called a slime coat. Now, not all bacteria have these, but a capsule or a slime coat is basically that. It's a layer of a slimy-like material that covers the bacterium, and it plays a couple of roles. It can help the bacteria form what's called a biofilm, so help the bacteria all kind of stick together and prevent them from drying out. So they form a thin sort of film layer on the surface on some sort of surface. But the capsule or slime coat can also play a very important role in helping the bacteria to hide from our immune system, so to avoid immune detection. So some of the bad bacteria, the ones that cause diseases, have a slime coat or a capsule that makes it that much more difficult for our immune system to actually detect that they're there. And the reason for that is our immune system normally detects proteins and things that are in the surface of the plasma membrane. So by having that extra layer of kind of slimy material on the outside, it just makes it more difficult for our immune system to see them. And lastly, there's a flagellum. Again, not all bacteria have flagella. So flagellum is one, or flagella is several. And some bacteria have none, some have a single flagellum, and some have lots of flagella. And these help the bacteria essentially to move, and so it's kind of, I believe it actually sort of moves in a little bit of a corkscrew kind of motion, and it helps to propel the bacterium forward. You can get what's almost like a smaller version, sort of shown here. Sometimes these are called pili, and they can be involved in movement of the bacteria too. So those are the parts. Now let's look at bacterial growth. So bacteria love warm, moist conditions, and that's why the body is actually a perfect place for bacteria to grow. It's warm, it's usually moist, so it's full of nutrients. It's got all the things that bacteria really need to grow. So one way to reduce bacterial growth is reducing the temperature of something by 10 degrees, basically halves the growth rate. So that's one of the main reasons we put things in the fridge. If we look at the bacterial growth, bacteria reproduce asexually. That means they don't need to have sex, they don't need a partner, there's no male or female, they simply grow by what we call binary fission. They basically grow in size, and when they reach a certain size, they'll divide or split in two. They'll make a copy of their DNA first, so that once they've divided, each bacteria has its own set of DNA. But essentially, they'll grow and divide basically just by mitosis. So binary fission means that they get larger, 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 and then they split in two, and you've got two exact copies that are genetically identical. And some bacteria can actually divide as often as once every 20 minutes. And that means the numbers actually can double every 20 minutes. So if you start with one bacteria, after 20 minutes you'll have 2, after another 20 minutes you'll have 4, and then 8, then 16, then 32, then 64, 128, 256, 512, 1024, 2040, and the numbers start to get really big really quick. And that's how you can actually have millions and millions of bacteria after several hours. They can get up into the millions. And if you graph it, the bacteria numbers look like this. They sort of steadily skyrocket upwards, and this line gets more and more, looks more and more like it's going straight up. So we call it exponential growth. Bacteria, under the right conditions, can grow exponentially. Now, whatever's going on, they can't actually keep growing exponentially forever. So eventually, bacteria will actually run out of food or space, or the buildup of waste products will accumulate, or they may be eaten by other organisms, so like amoeba or something like that. So at some point, something will limit their growth and the numbers will actually level off. So you can see we call these limiting factors. So they might run out of water or moisture, or they might run out of an energy source or some nutrient. One factor that's commonly a limiting factor for bacteria is iron. A lot of foods aren't rich in iron, and some bacteria need iron to grow. growth, that can be a limiting factor. Space, they can even simply run out of space, or the build-up of waste products, or the conditions can become too acidic or basic, and that's often actually due to certain waste products that are being produced by the bacteria. So the build-up of certain waste products, or if the environment becomes too acidic or basic, that's going to limit the number of bacteria too. So when we talk about bacterial growth, we say there are 4 phases to a typically to bacterial growth. And let me just grab a pen. So the first one is what's often referred to as the lag phase, where the line is fairly flat. And this is where the bacteria, so usually we graph the number of bacteria down the left, and along the bottom we'd have time along the bottom, just like my graph in the corner there. So the first phase is what we call the lag phase, where bacteria grow really slowly, and essentially that's even if you put bacteria in perfect conditions, they have to first adapt slightly to those conditions. So the lag phase, the bacteria are adapting to the new conditions, and what that essentially means, for instance, let's say there's an abundant supply of a certain nutrient or food source. Well, before the bacteria can start actually using those nutrients or those food sources, they're going to have to start making some enzymes. Just like we use enzymes in our stomach to break down food, they're going to have to start to produce enzymes to actually break that down. So once they've started to produce enzymes to break down the food, they then have to sort of do some of that digestion and absorb those nutrients, convert them to energy, grow larger in size, and then start copying their DNA, all those sorts of things. And then they can start dividing and continue to grow and divide. And that's when we then get the second part of the graph. The second phase is what we call the exponential or the log phase. And that's where we get a very rapid growth, where the growth rapidly increases. Then the third phase is what we call the stationary phase. So we had the lag phase, and then the log or the exponential phase. Then we've got the stationary phase, and so that's where the numbers start to level off, where this line becomes flatter again. And that's due to these limiting factors. So at this point, limiting factors are limiting the maximum number of bacteria. And then we go into what's either called the decline or the death phase, where often the bacterial numbers will actually start to drop off again. And this is because, again, just the build-up of wastes or the lack of a nutrient, if it really starts to run out, the numbers can actually start to drop off and they'll die. So if you put the bacteria, for instance, if we were growing bacteria in a jar, a limiting factor here, what limits the maximum number of bacteria, could be the amount of food that was in the jar. And so if you put more food in the jar, the graph will show a similar shape, and it will just go higher. They can support a larger, but eventually they're still going to run out, and then the numbers will drop off again too. So if you give them more food and space and moisture, essentially you'll get a higher point there. If you increase the temperature, if you make it hotter, slightly warmer for the bacteria, that affects how fast they can grow. So if it's slightly warmer, that often means bacteria might grow quicker. I mean, if it's too hot, you'll start to kill them. But if they grow quicker, you'll start to see a steeper line earlier, and then it will still level off at the same point. So still reaching the same maximum if you've just increased the temperature, but it goes up a little bit quicker, a little bit earlier. If you cool them down, if you put it in the fridge, essentially they'll grow a bit slower, so it'll take longer for that increase to occur. But again, you'll still reach the same maximum, and then eventually level off, because there's only so much food in the jar, and then it'll drop off a little bit later. So essentially, if you give them more food, you increase the maximum, so the graph goes higher. If you increase the temperature or change the temperature, you increase how fast, the time, so the graph shifts left or right. So it's the number of bacteria, the maximum number of bacteria is changed by changing the amount of available water and food and things like that. So that shifts the graph up and down. The number changes the number of bacteria, whereas things like temperature just change the speed at which they grow, so that changes the time left and right. It shifts the graph either left or right. So that's bacterial growth. So we had the lag phase, the slow bit at the beginning, adapting to the new conditions, making enzymes to break down the available food source, those sorts of things. The log phase, or the exponential phase, where we get rapid growth because there are no limiting factors yet. Then the stationary phase, where it levels off at the top, limiting factors starting to limit the number of bacteria, and then the death or the decline phase, where one of these limiting factors actually starts to reduce the number of bacteria. Cool. All right. So getting there towards the end. Two other points. Bacterial growth. Bacteria can be considered either autotrophs, which means they make their own food. And usually through photosynthesis, so like plants. So these are plant-like bacteria. They're producers. They make their food through photosynthesis. And the best way to remember that is autotroph. They're kind of automatic. They make their own food. Whereas heterotrophs are what we call consumers. They have to break down living or dead material, organic material, that's already been made by some other living organism. So they eat basically living or dead material. So these are basically the consumers, or more like the animal type of bacteria. So bacteria aren't plants or animals, but similar to plants are considered producers and animals are consumers. We've got a similar classification for bacteria. They're either autotrophs, producers, or heterotrophs, consumers. Alright, so one question that sometimes comes up, or a student might ask, is how does a bacteria feed on things that are much bigger than itself? So if I leave this lovely looking hamburger out on the bench, eventually it's going to start to grow bacteria and things like fungi too. So how can a bacteria that is much smaller than it's even shown here, that is totally microscopic, feed on something that is much bigger than itself? Well, the answer is basically this. It's called extracellular digestion. Extracellular digestion. And it basically involves just a couple of steps. A bacteria secretes enzymes. Those enzymes break down the food into particles. So digestive enzymes degrade or break down the food substance. And that releases nutrients, which can then be absorbed by the bacteria. Because they're small enough, they can be absorbed through the membrane and are taken up by the bacteria and used as building blocks and energy and an energy source and things like that. So extracellular digestion means basically enzymes are secreted onto the food. They break down the food into small food particles or molecules into their chemical components, and those are what are actually absorbed. And so bacteria aren't the only species that do this. We also, apparently spiders, some spiders do this. So they wrap their prey in a small cocoon, and then they basically inject into that with their fangs digestive enzymes, which break down their prey, and then they basically suck up what's sort of like a soupy mixture, and they absorb the nutrients from that. So the digestion doesn't occur sort of in a stomach inside them. It is actually occurring outside of them, and that's what the term means. Extracellular, outside of the cells, or outside of the organism, digestion. So the digestion is occurring outside. So bacteria feed by extracellular digestion, secreting enzymes onto the food. They break it down and then absorbing those chemical components, those nutrients, into the bacterium. So there you go. That's basically a definition there for extracellular digestion. Alright, lastly, bacteria can either grow anaerobically or aerobically. So anaerobic growth means that some of them can grow without oxygen, and they get their energy through this process without oxygen. And this process, anaerobic respiration, is sometimes also referred to as fermentation. So some can grow without oxygen, whereas others grow aerobically, undergo aerobic respiration, which means they need oxygen to grow. So they obtain their energy by respiration. Both of these are technically types of respiration. We say anaerobic respiration and aerobic respiration, but sometimes sort of more colloquially we refer to this as fermentation, and usually when we talk about respiration we normally are referring to aerobic, although technically anaerobic, this is actually anaerobic respiration as well. We'll look a little bit more at respiration, how that works later. Lastly, there you go. This is, you sort of maybe thinking what's going on there. This is those pili, those thin tube-like structures that can be involved in propelling bacteria. They can actually form connections, little bridges between the bacteria. And this is what's sometimes referred to as bacterial sex. And it's not technically sexual reproduction, but what can happen is small sections of DNA. So apart from that chromosome, the main loop of DNA, some bacteria actually have small loops of DNA. Some bacteria actually have small loops of DNA, which are called plasmids. So they're just a small loop, much smaller than the rest of the DNA or the chromosome. It's a small circular piece of DNA, and often they contain genes. Some of them, unfortunately, contain genes that make bacteria resistant to antibiotics. So through making these little bridges, these pili, which can connect to bacteria, a plasmid can be transferred from one to the other, or a copy of the plasmid can be transferred from one to another, and that's actually how bacteria can gain can become resistant to drugs like antibiotics and things like that. So bacteria can technically exchange some DNA too. Lastly, we mentioned this briefly in the last video, a lot of bacteria are named based on their shape. So if they're rod-shaped, they're a type of bacillus. If they're sort of bean or bent-shaped, they'll often be named Vibrio something. Spiral-shaped, Spirillum, or round, Coccus. So a good example, like meningococcal meningitis is a round bacterium. And so I've got a few pictures here. You can see here under the microscope using a special dye that makes them fluoresce, kind of glow in the dark. Some of those cocci, those round bacteria. These are actually tiny rod-shaped bacteria that are actually infected inside a human or an animal cell. So the animal cell, you can see that these big splodges here are the nucleus, the control center of the animal cell, and these little green kind of things here are small rod-like bacteria that have actually infected inside that cell. This is an E.Coli, and you can see it's got flagella, so commonly found in our gut. Another type of E.Coli, this is one that actually is, so most E.Coli don't actually cause us any problem. This one gives you really bad kind of diarrhea. You'd probably even have bloody kind of diarrhea with this. Helicobacter pylori is a type of Vibrio bacteria. Again, causes, I think, stomach ulcers and things like that. Another Helicobacter pylori. Listeria monocytogenes causes listeria. Mycobacterium tuberculosis, you may have heard of, causes tuberculosis or TB. Another rod shape. Listeria meningitidis, a bacteria that's a cocci, so you can see, and it forms long chains of them. So some of these bacteria form long chains or can be linked together. Staph aureus is another cocci, a round type of bacteria, and this causes what's often referred to as school sores, which are often because they occur in younger people, and they're sort of small pussy sores that form in the skin. So there's a few pictures of bacteria, and those are, I guess, the basic characteristics or the key aspects of bacteria. So I hope you found that helpful, and we'll start having a look at some of the other types of microorganisms.